The Stem Cell Network NRW is an initiative funded by the state of North Rhine-Westphalia. Through its centers across NRW, it covers the broad spectrum of scientific research issues in and around stem cell research. RWTH Aachen University is one of the leading engineering universities worldwide, and therefore the link between stem cell research and engineering takes on a special significance. It also involves a strong element of applied research, for example in tissue engineering and bioprinting.
At RWTH Aachen University, we work with several stem cell types: blood stem cells, cartilage and bone stem cells, neural stem cells, and embryonic stem cells. And we also work with engineered stem cells, such as induced pluripotent stem cells or iPS cells.
At RWTH Aachen University, leading stem cell researchers investigate the so-called biointerface, where cells are combined with biomaterial scaffolds in specific bioreactors to produce viable 3D constructs for future therapies in the clinics. In the research group, mesenchymal stem cells, known as MSCs, are isolated, expanded, and differentiated from bone marrow, the umbilical cord matrix, and placenta tissue. Mesenchymal stem cells have a positive effect on wound healing processes in the body and are involved in regenerating and modulating the structures surrounding the cells.
Our research is focused on guiding mesenchymal stem cells out of the niche in the body towards a tissue defect in order to improve the body's endogenous regeneration processes. And for this reason, we use biomaterials loaded with specific growth factors. In addition, we focus on stem cell-based tissue engineering, and here we concentrate on bone replacement strategies using mesenchymal stem cells and suitable biomaterials. And these can be metals, ceramics, or polymers.
Even though standardized bone replacement is still a long way off, the research group has successfully carried out preclinical trials in large animal models. Here, support materials were loaded with mesenchymal stem cells, differentiated into bone cells in bioreactors, and used in preclinical studies. The results are promising and show excellent osseointegration.
Although all the technical components can be used in a highly standardized and reproducible manner, we are limited in how we can standardize and predict the behavior of the cells. And therefore, it's not easy to predict the long-term behavior of tissue engineering constructs in patients. We are currently facing these challenges on the prediction of the behavior of endogenous cells in the production process.
Modern 3D printing technologies make it possible to generate spatial tissue constructs, created individually for each patient. These 3D printing technologies, known as bioprinting, are being developed in the Division of Dental Materials and Biomaterial Research.
On account of the lack of available donor organs and donor tissue, and the clinical problem of possible rejection responses to implanting foreign tissue, in the field of tissue engineering, research teams worldwide are intensively researching the possibility of creating replacement tissue in the lab.
In bioprinting, endogenous cells are embedded in a carrier fluid. Via printing heads specially designed for the sensitive cells, the cell suspensions are printed drop by drop and then stabilized post-output via a chemophysical process. Using a patented technique, it is also possible to create larger three-dimensional replacement structures. The cell structures then mature into functional replacement tissue when the 3D constructs are placed into special bioreactors. As well as their potential use as implants in patients, however, 3D printed cell constructs can also be used according to the organ-on-a-chip principle. Here, in vitro tissue models are used to solve fundamental biological, medical, and pharmacological questions. That has a great deal of potential as screening platforms for new medication in the pharmaceutical industry.
The use of bioprinting to produce replacement tissue as an individual patient implant is still largely at the research and development stage. Printing an entire organ, for example, an artificial liver, will probably remain a utopian ideal for some time to come. For less complex tissue structures, however, for example, partial skin replacement for burns victims, bioprinting technology could open up new possibilities for clinical treatment before long.
The Division of Stem Cell Biology and Cellular Engineering is investigating the molecular mechanisms which control stem cell functions. To this end, they are particularly examining the stem cells from human tissue responsible for lifelong regeneration. By reprogramming stem cells, the epigenetic patterns which appear as part of aging and cellular differentiation are completely erased. These stem cells really do seem to be rejuvenated. They can be cultivated over many months without showing signs of aging. Furthermore, the telomeres, the protective caps at the ends of chromosomes, lengthen again during reprogramming.
We are now able to better understand the developmental program of stem cells and how this is changed during differentiation. And we're also using this knowledge of epigenetic changes to establish biomarkers, to track diseases and to better understand the process of aging of the organism.
Interestingly, this age assessment seems to reflect the donor's biological age rather than their real chronological age. In addition, influential factors such as obesity, smoking, and diet increase epigenetic age and thus seem to directly influence the aging program of the cells. Scientists are continuing to work on epigenetic biomarkers, which should help us to gain a deeper understanding of diseases and the aging process. The Institute of Cell Biology works on which molecular switches control the growth and differentiation of cells. The goal is the identification of genes, factors, and conditions to direct the development options of stem cells. This also includes the generation of cells with new and desired engineered characteristics. These studies are carried out with hematopoietic stem cells, embryonic stem cells, and iPS cells. A particular focus of the Institute is on a highly specialized leukocyte population referred to as dendritic cells. With their dendrites, dendritic cells detect pathogens, such as bacteria and viruses, and activate the immune system.
A further focus of our research is on diseased blood cells, such as leukemia. Leukemic cells harbor specific mutation which cause the disease, and we employ iPS cell technology to isolate and study leukemic stem cells with these mutations. And we use iPS cell-derived cells for drug screening, and this allows us to devise treatments to patients that are specifically tailored to the respective disease.
The Division of Neurophysiology examines stem cell-derived nerve cells. Using an innovative technique, induced pluripotent stem cells from chronic pain patients are converted into human nerve cells in the lab. These carry the genetic code of the persons affected and thus allow mutations and other errors to be examined directly in these cells. In particular, the researchers in Aachen are using cells from patients with pain syndromes caused by genetic factors. This enables them to replicate the cellular changes, especially in terms of electrical excitability.
Patients with chronic pain syndromes, such as erythromelalgia, suffer from unimaginably severe pain. They describe the pain during attacks as feeling as if someone were pouring hot lava over their skin. To be able to investigate the cellular processes which underlie this and other kinds of pain, we have reprogrammed skin cells of these patients into stem cells. With the aid of substances that change certain signaling pathways in these cells, we are now able to produce neurons from these stem cells which react to painful stimuli in the skin, for example, to heat.
Information about potentially painful stimuli is passed via electrical signals along the nerve cells to the brain, where they are then detected. These electrical signals can be measured with the patch clamp method. Very fine electrodes are placed directly onto the cell bodies of the neurons, and the electrical activity of the nerve cells is then measured.
Many mutations that lead to chronic pain are found in the gene of the voltage-gated sodium channel. This is a protein which is key for generating electrical signals in the nerve cell. If a patient carries such a mutation, this often leads to the nerve cells reacting to very weak stimuli. We are able to show that this hyperexcitability in the patient cells is quite marked.
This study, and others like it, suggests that the sodium channel plays a very central role in human pain perception. If we were able to specifically modify this channel, then it is probable that we would also be able to help patients in pain. The researchers in Aachen are using a spider toxin, with which they have already been able to successfully influence the cellular changes in the neurons of patients.
Naturally occurring toxins are a good instrument for paving the way towards the development of better pain medication. Unfortunately, we cannot really use them in humans, which is why we need to develop better medications that specifically inhibit the neurons that transmit the pain signals. The neurons derived from iPS cells now provide us with a human system, which will hopefully support the development of new and improved medication for the patients.
As a production engineering research institute, the Fraunhofer IPT has many years of in-house experience in production processes. Therefore, experience acquired in past and ongoing projects is used in which fully automated platforms for the production of induced pluripotent stem cells or mesenchymal stem cells for research and therapy have been developed.
Cells, in particular patient-specific cells, are inherently individual and display this individual behavior during stem cell production. And controlling this behavior is a challenge which we need to meet. During the production process, we continuously measure specific cell parameters such as cell growth and differentiation, and then we adapt the operation of the production facility accordingly.
The goal is that stem cell production runs fully automatically. The mastery of biological variability can be understood primarily as a measurement challenge. Adaptive control mechanisms can be established by using the measurement data, thus enabling a fully automated production of homogeneous patient-specific cells.
So we can take full advantage of this production technology for stem cell research, and we can produce stem cells in high cell numbers under standardized conditions on an industrial scale. The production processes are run in parallel so that we can produce stem cells and cells derived thereof for a large number of patients simultaneously.
The stem cell network NRW regards itself as a catalyst for research and innovation in sustainable developments in the field of personalized medicine, developments which are becoming increasingly important in view of demographic change. In this regard, it is a prime example of the NRW government's concept for identifying and funding future research fields, creating a common platform for all those involved for the benefit of research, industry, and above all, for society.